Hello friends, this is Dr. Sachidanand Singh from Faculty of Biotechnology. So, in my previous lectures, you have understood about the techniques and terms related to protein modeling and structure prediction, and it is the module from protein modeling and analysis system in SWAM module. So, now let us see in this module what we are going to see, or in this lecture series. We have already performed n number of structure prediction method. Now, let us see another software known as Geno3D. It is also a modeling software. So, now let us learn within this lecture about Geno3D modeling server and its all the practical applications, experiments, results, discussion, challenges, advantages and all those stuff. So, let us start with this. So, Geno3D is a modeling server platform. A detailed explanation of Geno3D protein modeling software, which will include its application, methodology, algorithm, advantages, disadvantages, and also we will try to understand with an example. So, Geno3D is a protein modeling software. So, let us see its comprehensive overview. So, to understand its comprehensive overview, we will just have a brief introduction about protein structure prediction. So, what is protein structure prediction? Proteins are the essential macromolecules in living organisms, performing a wide range of biological functions. Understanding their three-dimensional 3D structure is crucial for elucidating their functions and potential applications in drug discovery, disease research and biotechnology. However, experimental determination of protein structures can be time consuming and costly. This is where computational methods like protein structure prediction comes into play. Application of Geno3D. Geno3D is a bioinformatics software tool designed for protein structure prediction. Its primary application is the computational protein modeling of protein as 3D structures based on their amino acid sequences. This technology has widespread use in various domains, including, let us refer few of them. Drug discovery. Predicting protein structures aids in understanding how drugs interact with specific targets, enabling rational drug design. Second is functional annotation. Geno3D can help in annotating the functions of protein with unknown structures by inferring their structures based on homology to known proteins. This functional annotation can also be done through Geno3D. Third one is biological research. It plays a vital role in understanding protein functions, interactions and biological pathway. Geno3D helps in biotechnology applications as well. Protein structure prediction supports the design of novel enzymes and protein engineering disease mechanism study also. Specifically in disease mechanism study, also Geno3D application is used. It contributes to studying the molecular basis of diseases and identifying potential therapeutic targets. Now, let us see in brief about methodology used in Geno3D server. Geno3D utilizes homology modeling. We all know homology modeling is the three-dimensional protein structure algorithmic approach in which more than 30% identical sequence as a target should be reported. Homology modeling approach to predict protein structure is used in Geno3D application. The process involves several key aspects. Let us see one by one. Template selection. Geno3D searches public databases for proteins with known 3D structures that are homologous, structurally similar to the target protein. Second, sequence alignment. the amino acid sequence of the target protein is aligned with the sequences of the selected template protein, which are 3D structure protein. This alignment identifies corresponding residues between the target and the template. So, template are what? Your 3D structure proteins. Third concept is model building. Based on the alignment information, Geno3D constructs a 3D model of the target protein by copying the 3D coordinates from the template structure and adjusting them according to the alignment. Fourth one is model refinement. The generated model may undergo refinement steps to optimize geometry, eliminate steric clashes and improve overall quality. The last is validation. The final model protein is assessed for quality and compatibility with experimentally data if available or been compared. Now, let us see algorithms used in Geno3D. So, the algorithm used in Geno3D are: Geno3D employs a combination of algorithm to perform its task effectively. So, there are different algorithms. These include: first, sequence alignment algorithm. Geno3D uses algorithms like BLAST. What is BLAST? Basic Local Alignment Search Tool. It helps in identifying homologous sequences in databases. Second one, homology modeling algorithms. The specifics of the homology modeling algorithm may vary depending on the version of Geno3D used. So, there are different versions. As the algorithm enhances, the software also give the better enhanced result. Common algorithms including Geno3D are modeler and Swiss model. Now, let us see few advantages of Geno3D. Geno3D offers several advantages that make it a valuable tool in protein structure prediction. First one is reliable predictions. When suitable templates are available, specific 3D structure is available, Geno3D can produce high accurate 3D structures. The unknown sequence can be reported as high accurate 3D structures. Second, very important, rapid modeling. It can generate 3D models relatively quickly compared to experimental methods, accelerating research and drug discovery at a greater scientific efforts. Third one, functional annotation. Geno3D can annotate the function of proteins with unknown structures by inferring their structures based on homology to the known protein. So, homologous based approach is been taken care to understand the function or structure of the protein. Last one is cost effective. It is a very cost effective technique of computational modeling which helps in comparing the experimental methods, and it can be a resource intensive approach. What are the disadvantages or challenges of Geno3D? Despite its advantages, Geno3D has limitations and potential drawbacks or challenges. First one, dependent on template availability. The accuracy of Geno3D prediction heavily relies on availability of closely related template structures. Second one, lack of suitable template can reduce the reliability of the protein structure. What is the second challenge? Limitation of novel proteins. Geno3D may struggle with proteins that lack close homologs in existing databases. What is the third challenge? Quality variation. The quality of prediction may vary depending on the quality of the alignment and template structures used. What are the different? Now, let us see the different examples of Geno3D for usage and application. Let us illustrate how Geno3D can be applied with an example. Then later we will see with the server on set by set. Imagine we are a researcher studying a novel protein from a pathogenic microorganism. This protein is a crucial for the microorganism survival. So, by understanding or literature survey, a researcher has identified for this microorganism, this protein, which is a novel protein, is crucial for its survival, but its 3D structure is not known. So, this can be seen from the UniProt database. Now, we want to understand the structure. So, this question is arised in the researcher or the student or the scientist. Now, let us start doing a 3D structure prediction. Now, we want to understand its structure to potentially design drugs targeting this protein, so that the survival can be stopped. So, for this, the first thing will be the input sequence. So, you as an input the amino acid sequence of the novel protein into Geno3D. So, now the sequence which we have, we can retrieve from n number of different databases of protein, and we can give as an input into Geno3D server. Now, what Geno3D will do? It will do template search. Geno3D searches public databases for proteins with known 3D structures, and known 3D structures they will retrieve from where? Yes, protein data bank, PDB. So, protein with known 3D structures that are highly similar or homologous will be given as a hit for the template search. Alignment, the next step will be alignment and model building. The software aligns to the protein of the interest sequence with the sequences of the selected template proteins. If the construct a 3D model of the protein by adopting 3D coordinates from the template structure. Now, the protein modeling will initiate by seeing the coordinates of the template, and the 3D protein will be developed for the sequence which we have given as an input. Model refinement, final after the model building, model refinement will be performed. The generated model will undergo refinement to improve its quality and to ensure that it does not violate the known principles of protein structure. After model refinement, validation will be performed. Under the validation, the final 3D model will be evaluated for its quality, and if available, we might compare it to experimental data or to use it for virtual screening of potential drug compounds. So, this example demonstrates how Geno3D can provide a valuable insights for constructing a 3D structure of a novel protein, potentially which can be facilitated for drug discovery efforts. So, Geno3D is a valuable bioinformatics tool for protein structure prediction. Once used a Geno3D to predict the 3D structure of a protein, the results typically come in different formats that allows for further analysis and interpretation. So, the protein structure identified through Geno3D need to go for another steps. So, it gives us different formats. So, this was a concise overview and a brief introduction about Geno3D protein structure prediction analysis. Now, let us see Geno3D protein structure prediction results, analysis, and what type of formats it gives. These format helps Geno3D input to give as an output, and these outputs can be utilized in the further steps. So, as we saw in the previous steps that how Geno3D can be used, now let us see what are the output formats of Geno3D. So, the very first one is PDB format. One of the standard format in which Geno3D provides protein structure prediction is PDB format, that is also known as protein data bank format. The PDB format contains detailed information about the 3D coordinates of atoms in the protein structure. Each atom's position is specified along with its associated atom type, residue number, and chain identifier. So, these are the these are the inputs present in PDB. This format is widely recognized and can be used for visualization and further analysis in various molecular modeling software. It is used in different softwares such as PyMol, VMD, and Chimera. Second one is how to visualize it. So, for visualization, One of the primary way to analyze Geno3D result is through visualization. So, by visualization only the protein structure can be seen or analyzed. Researchers can load the predicted protein structure in PDB format in molecular visualization softwares to examine its 3D confirmation. Visualization helps to identify structural behaviors, structural features such as secondary structures, specifically alpha helix and beta sheets. It also helps in identifying active sites and potential binding pockets in further responses. Researchers or scientists can also compare the predicted structures with experimental structures or to know functional domains or motifs. Another format or approaches or results which we can be identified is Ramachandran plot. In previous lecture also a very clean and neatly explanation was given about Ramachandran plot. The Ramachandran plot is a crucial tool for assessing the quality of a protein structure prediction. It provides insights into the dihedral angle, that is phi and psi, of amino acids in the protein. Geno3D results often includes a Ramachandran plot representation as well, which can help to identify regions of the structure that may require further refinement, and this further refinement is also known as step by step loop modeling process. The plot should ideally show most phi and psi angles in the favored and allowed region, indicating a reliable prediction. This whole representation of Ramachandran plot is already explained in the previous lecture. Kindly refer those lecture back for more detailed understanding. The next results may be as RMSD analysis, that is root mean square deviation. What is root mean square deviation? It is a measure of structural similarity between the predicted model and a reference structure, such as an experimentally determined crystal structure. Geno3D users can perform RMSD analysis to quantitatively assess how well the predicted structure aligns with a known structure. Lower RMSD values indicates a better structural match or mapping. So, this also example of RMSD was given in the first introductory lecture of protein structure prediction and analysis. The next result may be quality scores. Geno3D often provides quality scores or assessment metrics method for the predicted structure. These scores reflect the overall reliability and accuracy of the model. Common matrices includes a GA341 score and Q mean. Higher scores generally indicates a more trustworthy prediction method for protein structure. The next results or upcoming portion may be ligand binding site prediction. In some cases or approaches, Geno3D may also predict potential ligand binding sites within the protein structure. Identifying these sites is a crucial for understanding the protein function and potential interactions with small molecules or other proteins. Researchers can use this information for virtual screening and drug design studies. The another basic approach and results may be function prediction or application of Geno3D. While Geno3D primarily focuses on structure prediction, researchers can also use the predicted 3D structure to infer the protein's function. Structural features such as active sites, binding pockets and structural motifs can also provide clues about its potential role in biological process. Another application may be comparative analysis. Geno3D results can be further analyzed through comparative modeling and structural bioinformatic techniques. Researchers or scientists may compare the predicted structure to existing structures in databases, perform sequence structural alignments and explore evolutionary relationships. This analysis can lead to insight into protein evolution and functional characterization. It may also help in model refinement. If the Geno3D prediction is considered preliminary or less accurate, researchers, scientists or students may choose to refine the model using molecular dynamic simulation, which also has been introduced you in the previous lecture. Kindly refer the same for molecular dynamic simulation basic understanding. So, for the refinement, MD simulation may be again utilized for further refinement algorithms. Refinement will aims to improve the model's overall quality and reliability. So, in conclusion of results and discussion and output, Geno3D protein structure prediction results come in a format that facilitates comprehensive analysis. Scientist and student can visualize the predicted structure, assess its quality through metrics like RMSD, root mean square deviation, Ramachandran plot, exploring potential functional insights. Additionally, Geno3D users can compare their predictions to experimental structures and perform further analysis to advance their research in various fields, including drug discovery, molecular biology and structural biology. So, this was all about Geno3D application, results, discussion and input and output structures. So, this was a detailed understanding of Geno3D. So, now let us see in a brief and a concise way how a Geno3D web server looks like, or how a Geno3D can be used with a example on the web portal. So, the web portal or URL of Geno3D is http://geno3d-pbilibcp.fr. It is the web server name for protein molecular modeling. So, we can use this URL and we can type into the Google. After that, we will receive the Geno3D web page, and it is a web server for protein molecular modeling. Then Geno3D web page will be opened. Geno3D is a web server which is provided and developed by PBIL Loyan Garland team. It uses homology modeling approach. We can see the whole overview. It predicts 3D structure from amino acid sequence. The most important aspect of Geno3D server is it helps in evolutionary similarity. It is developed by Swiss Institute of Bioinformatics, in short known as SIB. So, why we use this Geno3D we have already shown, but in a practical application when there are lack of solved structures, then starting point of structure and functional study is not been reported. It helps in predicting the orthologous proteins as well. It helps in structural membrane protein identification. It helps in developing or constructing large and complex protein. Very important example shown as a help file of Geno3D also is to construct protein like G protein coupled receptor GPCRs and cytochrome P40 are the excellent example of giving a utility of homology modeling. So, Geno3D uses homology model algorithm which can serve as useful guides for designing mutagenesis experiments, ligand binding studies and functional analysis, providing valuable insights into protein behavior. So, as we said Geno3D can be available into this portal. So, there are no restrictions on its use by non-profit institutions as well. Usage by and for commercial entities requires a license agreement. We can also send an email to the given mentioned email for its usage. What are the limitations? Geno3D server releases to generate a model until 500 amino acid, and it considers those parameters. So, we can only generate model up until 500 amino acid. By default, the modeling process made use of databases and PSA 3D sequences at 100% homology from PDB. After the 3 run of the similarity search, SciBlast. What is SciBlast? Position specific iterative basic local alignment search tool program will be utilized. Generation of 3 model will be used as a step. The user when taking up using this page will provide a sequence to be modeled, which is compared using the SciBLAS. It is a part and a flavor of basic local alignment search tool. The method against a protein sequence database issues from PDB, and all entities and the structure will be retrieved. Next, the user as an us will select PDB entries as template for molecular modeling. So, the example which we have taken in this case, we can take 1 BEP, 1 CC and 1 Beck as can be selected as the template on the basis of E value, that is expected chance mutation, query coverage and the statistical parameters considered over here. For each template, Geno3D server will what it will do? It will first compute secondary structure prediction. And what are secondary structure prediction? That alpha helix, beta sheets, loops and coil. It will also display percent of agreement in secondary structure. It will also do repartitioning of information from template to query sequence. In case of selection of multi templates, the RMSD, what is that? Root mean square deviation between the templates on carbon alpha is also displayed. So, for the sequence which we have given as an input, what will be the results of modeling? The first will be molecular 3D model. So, the thing which we have received is model underscore 1 dot pdb. This will display and this 3D model will be given. So, as we said, we will be receiving 3 protein models, model underscore 1, model underscore 2 and model underscore 3. These all protein models can be visualized and analyzed into a open source software known as RASMOL, which is a freely available software. Now, let us see the output data in the Genome3D, which we can see. So, about the whole molecule, we can calculate or understand its energy. We can see Ramachandran plot. We can also see the report.txt file. In report.txt file or the txt extension of the report of that input file, we can analyze and that content can be written as a scientific article or explanation for results and discussion in any paper. What is the input of report.txt file? The about each chain. It will explain deviation, restraints, comparison with each template. It will also explain the report in different formats like for specific chain. Finally, template selection valid.txt file will also be there to understand which template is been selected and how it has been reported. These all files can be used, utilized, analyzed and can be reported. Now, let us see in archives of local analysis what will be there. It will be given as archive.tarzz format. So, this we have to see, we have to unwind and see what is there in that. So, by understanding the all the outputs, the protein model can be understood and information about the whole molecule can be taken in sense of information about the energy. Second, Ramachandran plot can be understood. Third, deviation between all the models can be understood and explained. Fourth, number of restraints can be deduced from utilizing the template. Number of restraints which aren't respected in models can also be violated. When a parameter is not satisfactory, the line is written in red color. So, that we can see in the segment if it is mentioned. Information about each chain deviation between models on this chain, number of restraints deduced from the template and number of restraints that aren't respected in model will also be reported. Deviation between this chain and the template used to build this chain can also be explained. Illustration of analysis tool, usage of the PDB files and PDB structure also will be explained in yellow color. It will also explain all the templates which has been taken and their significant functional annotation also will be reported. So, this was all about the output of Geno3D in the web server. So, in the whole presentation and in this lecture, we have seen the whole alignment technique of Geno3D as application, as its advantages, as its report and everything. So, this all conclusion. Now, let us see about Geno3D. So, in conclusion, Geno3D and protein modeling tools have revolutionized our ability to predict protein structures and their functions. These tools have a profound impact on drug discovery, biological research and biotechnology. The future of Geno3D holds exciting possibilities, including improved accuracy, handling of novel proteins, integration with experimental data and contribution to personalized medicine and interactome modeling. As the field continues to evolve, Geno3D is poised to remain a vital resource for researchers seeking for unravel the mysteries of the protein world. So, that was all about the conclusion of Geno3D. Now, let us see what are the benefits of Geno3D and who are the beneficial persons which can use this Geno3D. First is bioinformatics students and researchers. So, in bioinformaticians and researchers may utilize this Geno3D for different approaches like protein structure prediction. It will utilize Geno3D for protein structure prediction, and researchers who are interested in 3D structures of protein. It will enable the exploration of protein folding and the effects of mutation on protein structure. The second one are molecular biologists, functional analysts. Functional analysts will under molecular biology will use Geno3D to infer the potential functions of protein with unknown structure. This is particularly valuable for studying newly discovered genes or protein. Third one are drug discovery scientists, in which they will use as a target identifications. Scientists working in drug discovery can utilize Geno3D to identify potential drug targets by predicting the 3D structure of proteins involved in diseases. The fourth one may be structural biologists. So, structural biologists will utilize for structure validation. So, for structure validation, structural biologists can use Geno3D to validate experimental structure by comparing them to computationally predicted model, and this will aid in quality control and provides insight into structural variations. The fifth application people will be genetists, and they will use for functional annotation. So, genetists can employ this server to identify and annotate the function of genes with unknown functions based on structural homology and known proteins. The sixth one may be the core biotechnologist in protein engineering. They may use to design and engineer proteins with desired properties such as enhanced catalytic activity or stability. The later one may be computational chemists. So, computational chemist people will use for molecular docking, and they may use it with molecular docking simulations to study protein ligand interaction and predicting binding affinities. The further approaches may be used by pharmaceutical researchers for structure based drug design and target based drug design. The pharmaceutical industry people may help it to use for designing novel drug candidates based on their interaction with target protein. So, this was all the applications about the Geno3D and the people or the group of scientists or researchers which will use Geno3D as their application point of view. So, within this whole lecture we went through the whole paradigm of Geno3D application, its usage and its current scenario. So, in the coming lecture we will see the pipeline of the protein structure prediction also. Thank you so much.